hi. Good evening, everyone. Good evening, everyone. Mood one flying. Yeah, so you guys join fast. We start the session. So today we are looking at muscle tissue. Alright. Okay, just giving people a minute or two to join. I've been Muson Leonard, Nicholas, who's this? Neri. Also, you guys, waiting for you. Kindly join so that we start the session. Okay. Alright. We're gonna gift banda. Okay, so muscle tissue. We're doing muscle tissue. So muscle tissue is just a specialized tissue that can contract and produce movement. It's just a specialized tissue that can contract and produce movement. So in the body, it makes up to 40 or 50% of the body mass, and it is essential for movement. These are just the major function: movement, posture, or maintains posture, production of heat. Okay, so the muscle tissue, they are in three types. We have the skeletal muscle, right? Okay, we have the skeletal muscle, we have the cardiac muscle, we have the smooth muscle. Okay, so muscles, the classification one, I'm saying we have skeletal muscle, we have the cardiac muscle. When I say cardiac, they are only found in the heart, right? So the smooth muscle, and this one you should know how to differentiate them. These two, they have striations. These two, skeletal muscle and cardiac muscle, have striations. Striations. This one doesn't have striations, the smooth muscle, right? Okay. So now where can you find the skeletal muscle or skeletal muscle tissue? Those skeletal muscle tissue, you're going to find them, they will be attached to bones by tendons. They are also found in the facial muscles, the tongue, the pharynx, and the diaphragm. Do you know where the diaphragm is? The diaphragm. Okay. I hope today the network will be on our side. Okay. Right, let me give you the terminologies that you need to know. Okay, so when they say sarcolemma, what do they mean? Sarcolemma is just it's a plasma membrane. When they say sarcolemma, sarcolemma, we'll be exposed to terminologies like sarcolemma. Okay, so sarcolemma is just the plasma membrane. Two, we have the sarcoplasm. Okay, sarcoplasm is just the cytoplasm. Up to these, you can able to relate. We know the cytoplasm, we know the plasma membrane or the cell membrane. Yeah. We also know this terminology like sarcoplasmic reticulum. When they say sarcoplasmic reticulum, what are they referring to? So this is the endoplasmic reticulum. When they say sarcoplasmic reticulum. Okay, so this is just the endoplasmic reticulum. Okay, then when they say sarcosome, the sarcosome is just the mitochondria. The sarcosome is just the the mitochondria. Okay, I've talked about the classification of muscles. Now, what are the characteristics of the muscle tissue? What will be the characteristics? Let's talk about the characteristics of the same muscle. Okay. So number one, muscles, they will be able to excite. So muscles, they are excitable. They're able to fire an action potential. Okay. One, it's excitable. Of which you will dig deep into the excitability of these muscles when you start looking at the contraction of muscles in muscle physiology. For this one, I'm just giving you the pointers so that you get to know if you want to go and dig deep. But for anatomy, just know the structure and the function. That's enough. Yeah, and also some clinical relevance or clinical correlations. Yeah, for physiology, that's when you need to go and dig deep like that. So another thing, characteristic of the muscle tissue is that it's able to contract. It's contractile. It's contractile. Okay. So the muscle tissue is able to contract. The muscle tissue is able to contract. Okay. All right. For those of you joining, okay. So another characteristics of the muscle is that it's able to extend. It's extensible. It's extensible. So okay, let's look at this. The muscle in one extension. Then when the muscle this is the muscle. Okay. So this muscle, you are able to pull it. Get it able to extend. When you pull it, you're able to extend. That is extensible. Then it also has elasticity, like a rubber. It's able to extend like elastic. Yeah, these are the characteristics of the muscle. So to be able to extend. So when it extend, maybe it can be even look like flat, like so. Yeah, you are able to extend it. Okay, then the fourth one is elasticity. I've talked about it. Elasticity, you know the elasticity, like rubber, it's able to extend like rubber. Elasticity. Okay. Right. So let's talk about the skeletal muscle cell. The skeletal muscle cell. Okay. Skeletal muscle cell. So the skeletal muscle cell, it has what? It has the sarcoplasm. So these are the terminologies I give you initially to say when they say sarcoplasm, what do they mean? So sarcoplasm just means it's just the cytoplasm. Things that you really know, just the change of terminologies. So the sarcoplasm, just the cytoplasm. It also has the nucleus. It has the sarcoplasm. It also has the nucleus. For the sarcoplasm, that's where you will find what it has in the sarcoplasm. It has a dark A band. It has a dark A band, of which I will explain when I show you the structure of the sarcomere, what these dark band mean, and how they are located in the sarcomere, from which distance to which distance. Then it also has a light. This is dark, and it also has a light, which is the I band. Which is the I band. It has light which is the I band. Okay, and then for the nucleus is multinucleated. This one is multinucleated. Then it has flat and elongated. It is flat and elongated. Then it is peripherally placed. Okay, it is peripherally placed. Then the same skeletal muscle cell, it also has what? A connective tissue. The connective tissue. So I'm just giving you pointers. So this shouldn't limit you from going and go and research more, so that you know more if you want. So these the skeletal muscle, it also has a connective tissue. What are these connective tissue? One, we have the endomysium. Endomysium. Okay, that is the each fiber. That's one fiber, single fiber. Each fiber. That is the endomysium. Then we have the perimysium, which is the muscle fascicle. The perimysium. This is the muscle fascicle. Okay. This is the muscle fascicle. Right. Also of the epimysium, which will be the entire muscle now. The epimysium. This is the entire muscle. Okay. So again, I repeat, the major function of the muscle is able to produce movement, that is locomotion. It also maintains posture. It's able to stabilize joints, like I've talked about the skeletal muscles, how they are attached to tendons and whatever, to stabilize posture. But they also produce or generate heat. How is it generated? Like the way you are shivering, it's cold, like it has been rainy, now it's cold, you are shivering, so your muscles are able to generate heat. Okay, so that your temperature is maintained, okay, at a steady state, right? Okay, so now what can you talk about the histology of the skeletal muscle? So like I said, for both the cardiac and the skeletal have striations. So this one has striations. It also have a peripheral nuclei, like I've said on the pointers. Yeah. So these peripheral nuclei, they are located at the edge of the fiber, in the edges. Okay. So then the myofibrils, they are arranged in repeating units. So repeating units, what I'm saying, the myofibrils, that is, they are called the sarcomeres. So we'll look at the sarcomere, the structure of the sarcomere, which will be the functional contractile unit of the muscle. So one which functions in terms in contraction and relaxation of muscles. So I've talked about the function of the skeletal muscle, right? So these muscles, they are controlled, they are voluntary or somatic nervous system. So the motor neurons, they will release the acetylcholine at the neuromuscular junction. So this will trigger what? Contraction, of which will dig deep in physiology, in the muscle physiology. Then for the cardiac muscle tissue, they are located only in the heart hole, that is the myocardium. Get? Then the structure, it has branched fibers, it has single and central nucleus, sometimes there are two. It also has striated, like I said, if you are comparing it to the skeletal, and then cells, they are connected by intercalated disc. They are disc, what we call intercalated disc. So this disc, they they contain desmosomes. Desmosomes, they are the one which has a mechanical strength. It also has gap junctions, which are electrical coupling for synchronized contraction. Okay. Then the storage of the cardiac muscle, it has striations, like I said, the intercalated disc, which will appear as dark lines. It also has a central nucleus. They are rich in mitochondria. Do you know the mitochondria? Of course, you know. Yeah, which will be the powerhouse for, that is for high energy demand. Okay, the function of the cardiac muscle tissue. So this is involuntary. You don't have control over it. So like, yeah, for skeletal you have control over it, because you're able to say, okay, let me go this. Like, you're able to control your movement. For this one, it's involuntary. It's rhythmic contraction that pump blood. Yeah, like the way you can't tell the heart to say now pump blood. Do what, like, yeah. So these are the cardiac muscle tissue. Then the function of the cardiac muscle tissue, it also has the automaticity, which can contract without nerve stimulation due to pacemaker cells. It has cells which are called the pacemaker cells, right? Okay. It also has synchronization of contraction for efficient pumping of blood. So these are just the function of the cardiac muscle tissue. Okay. Then the smooth muscle tissue, you guys, you've learned, you've already covered the muscle tissue. I'm just learning through to give you the pointers to remind you. Okay. I can just say this is just a rapid view, of course, of which we've been sent the tutorial document. But stay with me, I'll show you the structure of the sarcomere. You need to understand the striding filament and how the muscle contract. Even the structures and the labeling of the sarcomere is very important. You need to understand that. Yeah, because the same sarcomere you'll find it in muscle physiology, of which we'll dig deep. Also know well the contraction mechanism of the muscle, both in skeletal and cardiac, also in smooth muscle. Alright, so where can you find the smooth muscles? Since I said the muscles, you've learned that muscles, they are in three categories: well, the skeletal, the cardiac, and the smooth. Now, the smooth muscles, smooth muscle tissue, where can you find them? So these are found in the halls of oral organs. Do you know when they say hollow organs? Hollow organs. So hollow organs like what? The blood vessels. So the blood vessel is a hollow organ. Okay. The blood vessels. The digestive tract. Okay. The GIT. The urinary bladder. These are hollow organs. The respiratory passages. The uterus and the reproductive tracts, the iris and ciliary body of eyes. That's where we'll find the smooth muscle tissue. Okay. Structure, it also has spindle-shaped cells. It also has single and central nucleus, and it is non-striated. This one is not striated. It's different from cardiac and skeletal. This one is smaller than skeletal fibers. You have to know this one is smaller than skeletal fibers. Okay. What will be the function of these smooth muscles? So these smooth muscles, the involuntary contractions. There is involuntary contraction. Okay. Then it also propels substances like peristalsis in GIT. They are in GIT. That's why I'm saying there are smooth muscle there, because when you swallow whatever food, yeah, there will be movement there, which are called in peristalsis. Peristalsis is also a powerful mechanism in physiology, of which you will learn in GIT physiology. Yeah, there's movement of food there. There's a very good mechanism in peristalsis. Alright. So these it's propel substance like in peristalsis and GIT, also in ureters. So in the other functions, it regulates lumen size. Okay. It's also able to regulate lumen size, that is vasoconstriction, vasodilation. Also controls the pupil size, the uterine contractions, all these things. Okay. So if you are behind, don't worry, I'll send the tutorial document so that you're able to learn through. Okay. So if you are with me, you can just give a thumb up. I mean, you can just like, so that see people who are with me here. Okay, we can now go to the sarcomere, which will be a very interesting part for this video. Okay, the sarcomere. Okay, I'm sure you saw this beautiful picture as we were studying your notes. So this is the sarcomere. Okay, this is the sarcomere. Okay. Right, so what do you understand by sarcomere? Hi, Gillespie. What do you understand by sarcomere? What is sarcomere in short? What is sarcomere? Okay, so sarcomere is just the basic function on the structural contractile unit of striated muscle. So these striated muscles what are these striated muscles that I said? So that is the skeletal and the cardiac. It's a basic functional and structural contractile unit of striated muscle. And the muscles that are striations, I said, is the skeletal and the cardiac, except the smooth muscle. Also, you can say it's just the repeating segment along a myofibril bounded by the Z disc or the Z lines. So these are the one shortening of sarcomeres called skeletal and cardiac muscle contraction. Okay. Now let's see the structure here. Now. Okay. Okay. Have you seen this big block here in the middle? So this big block, it has a name. So this one is what? So this is the thick filament. So this is called the thick filament. Okay. So this is the thick filament. So this is the thick filament. Okay. Then the middle one, the vertical line you are seeing here, this one. So this one is the M line. It's the M line. Do you know the protein that makes up the M line? Actually, there are many proteins, but there's one which is very important. So the protein which makes up the M line is called what? The myomesin. Okay, so you have a technical challenge here with the network, but don't worry, you'll be fully taken care of. So I'm saying the protein which makes up this vertical line you're seeing here, which is the M line, is what? It's the myomesin. It's the myomesin or myomesin. Okay, so the protein what? It's called the myomesin. Okay. Have you seen this zigzag line here? So this zigzag line, is called the Z disc, the Z disc or the Z disc. Okay. Do you know the protein which makes up the Z disc? So the protein which makes up the Z disc, it's called the alpha actinin. It's alpha actinin. Alpha actin. It's the alpha actin. The protein which makes up the Z disc is called the alpha actin. Okay. So since I say this one is a thick filament, actually the thick Today our network is not on our side, but I think you still manage. We will manage. Okay, right. Right. I hope you are following. Even though we have a technical challenge, you have to finish this thing. Okay. Right. So I said this thick filament. Since this is the thick filament, there is actually a protein which connects the thick filament. Okay, it connects the thick filament in place. So what is this protein called? What is this protein called? So this protein is called titin. So this is what? Titin. Titin. Okay. So this is called titin. Okay. Right. Then have you seen this line? Actually this line where there is this which is coiling like a helix. So there is this line. This line is what is called this line is the thin filament. This one is the thin filament. This line is called the thin filament. Okay. And I've seen this green line here. So green line is presenting the tropomyosin. The tropomyosin is representing the tropomyosin. Okay. And these things I've drawn here, they are not flowers. Actually, yeah, they are not flowers. Yeah, so these things is called the myosin head. It's the myosin head. It's the head of the myosin. Okay. So actually, the head of the myosin, it has three parts. There's head which act with the ATP and also the active site. It has two binding sites. Yeah, so it has also the neck. It also has the tail, which it contains of the alpha helix. Yeah, all those beautiful things. Right. So then the thing here we're seeing here, which is coiling inside in between the tropomyosin and the thin filament. So this is called the actin. Not the alpha actin. There is this. When you say the alpha actin is the protein which makes up the Z disc. So this actin. This is actin which binds the myosin head. Yeah, like when the muscle is there's excitation of muscle contraction, there will be this tropomyosin will stride out so that it will expose the binding site between the myosin head and the actin. Okay, of which I will explain the cross bridge. I'll give you the steps of the cross bridge in the same video. Alright. So what other thing do you need to know about this? Okay. Okay. So the region between, have you seen where the thick filament is ending? So the region between the same thick filament from this region to this region represent what? A certain band. Well, what is this band is called? This band is called what? The A band, or you can say it's the anisotropic. It's the anisotropic. Anisotropic. Okay. It's the central. The band, okay. The anisotropic. The A band. That's this region. You're supposed to draw this the sarcomere and know the labels. Okay, so I saw a certain question in physiology that required you to draw the sarcomere and also give the steps of the muscle contraction. Okay, so let's look at another thing. Another region or another band. Okay, Now, from where the today's network is very pathetic. Okay, so from the region where the thick filament is ending up to where the Z disk is. So from this region to this region. So this region is called the I band. This is the I band. So the I means, so this is the I band from the Z disk to where the thick filament is ending is called the I band. So the I means that is the isotropic band. Isotropic. Okay, that is the isotropic. Right, that is the isotropic. Okay, so on the same, let me zoom this one. There where there is topomyosin and also the actin and the right. So I'm saying where there is this, where there is topomyosin, also this helix of actin and the thin filament. When you zoom here, there's a certain protein which is called the troponin. Okay, there's a certain protein which is called the troponin. This troponin, it has 3 legs. Okay, I call it, it has 3 legs. It has this structure. Okay, it's like this, or 3 parts. Okay, let me draw it from the other side. Okay, I'll be using the same diagram. Let me just do it from the other side. Okay, right. So I'm saying the troponin, when you zoom from there, it has 2 sides. Okay, like this. It has this side. This side is where the calcium binds. Okay, and where the calcium binds is called, where the calcium binds, the site where the calcium is binding is called the troponin C. This is called the troponin C. And there's this side which is the topomyosin. This side is the topomyosin, and this other side is the actin. You see, yeah, in between those thin filament, there is topomyosin, and there's also actin. Okay, so where the site where the calcium binds is called the troponin C, the binding site of calcium. Now, what about the binding site of the topomyosin side? This is called the troponin T. It's not because the topomyosin starts with T, no. It is called the troponin T. Okay. Now, what about this side? So this side, on the actin side, is what? Troponin what? Is it troponin A? No. So don't take the first letters to say no, since this was troponin T, so because calcium starts with A, no, it's not like that. So this will be troponin I for the actin side. This will be troponin I. The I means inhibitory. Okay, so on the same sarcomere, like I showed you, to say these are the myosin heads. So the myosin head. Okay, now let's look into the structure of the myosin head. Okay, the myosin head. Okay, so this is the neck of the myosin head. This is 2 heads. There are 2 heads. So this head of the myosin has 2 binding sites. There's 2 binding sites. The one is for, another one is for ATP, and another one is for actinin, and the other one is for actin. Okay, so now the neck, this myosin head has the neck. So the neck, what are you going to find in the neck? The neck has essential right chain. There is essential right chain there. There's essential light chain, the ELC. There's also what is called the regulated light chain, the U regulatory light chain, the RLC. So these chains, they are the one which stabilizes the arm. Okay, it's like this, the hand. Now, yeah, it's the one which stabilizes. Like it will be during contraction, the head will be moving. To be moving up to bind to the active site and it cause contraction. Then it cocks back. It comes back to its normal position. Like these chains are the one which stabilizes the arm movement. You get, yeah. So the tail has what, the alpha helix. The alpha helix, just the protein. Okay, let me run through the pointers again. So on the sarcomere, like I said, the sarcomere, we have the thin filament here. The thin filament. So the thin filament consists of what? The double-stranded helix of the thin filament. This one has the double-stranded helix of the F-actin. There's F what? Actin. Actually, there is a protein that I've forgotten. Let me run through the show you the structure. There's a protein. Okay. Alright. That's when I've remembered. There's actually a protein connecting the thin filament to the Z disc. So you know, yeah, like this is the thin filament. Actually, the protein which connects this filament to this Z disc, this zigzag line, it is called what? The nebulin. Nebulin. It sounds like a good name, eh? The nebulin. Yeah, the nebulin. Right. So don't forget, the protein is the nebulin, which connects the thin filament to the Z disc. Now, the one which connects the thick filament to the Z disc is the titin. Okay. And the protein which makes up the Z disc is the alpha actin. The protein which makes up the middle line here, the vertical line, which is called the M line, is the malmesine. Okay. It's the malmesine. Also, you have to understand the region of the bands, the I band, the H band, also the A band, which is anisotropic, also the isotropic. Okay. Let's continue. So the thin filament consists of the double-stranded helix of the F-actin and actin-associated proteins. So what are these actin-associated proteins I'm talking about? Yeah, I'm talking about the topomyosin, the troponin, and the nebulin. Like I've said, the nebulin is the one which is connecting the thin filament to the Z disc. Simple, right? So this F-actin chain. This F-actin chain, it consists of two chains of the G-actin protein. It consists of two chains of the G-actin protein. So the G-actin protein is the globular protein, which each G-actin protein has an active binding site for myosin. And this active binding site remain covered. So it's like when there's no contraction, it will remain covered. Okay, so it will remain covered. It will remain covered to topomyosin protein chain during relaxation of the muscle. When the muscle is relaxed, meaning the topomyosin will remain covering what? The binding site for the myosin head. Okay, let me draw like this. The actin. Don't know. Welcome back. Okay, so this is like a simple structure I'm trying to illustrate. Okay, so the other thing, the thing you are seeing here is the thin filament, and this is the actin. Now here, that's where the active binding site are. This is the active binding. Oh, today network just not on our side. Okay, so yeah, like this is the thin filament on top, and this is the actin protein. And here I'm showing the active binding site, and these we are seeing here, these are the myosin head. So, and the line you are seeing here is a topomyosin. So during relaxation, this topomyosin, it covers the active binding. So the topomyosin covers the active binding site during relaxation. Okay, when the muscle is relaxing, during relaxation. Okay, so during contraction, this topomyosin is supposed to slide out so that it exposes this binding site, so that the myosin head can fit into the active site and cause what contraction of the muscle, like to cause the shortening of the sarcomere. Okay, so like the other head, since this myosin head has two binding sites, there is another one for ATP and another one is for actin. Okay. So when it fits into the active site, it will cause what the shortening of the sarcomere. Get? So that's the thing. Alright. So that's the thing I was saying, that the active binding site remain covered of the topomyosin protein during relaxation of the muscle. Okay, so now the topomyosin I'm saying is a polypeptide chain that lines parallel to the F-actin chain, and it masks or it covers the myosin binding site for the G-actin. Okay, now the important thing you're supposed to know is the troponin. The troponin is the key protein molecule for muscle contraction. When they ask you the key protein molecule for muscle contraction is called what? Troponin. Okay. So when they ask you what is the key protein molecule for muscle contraction, what will be your answer? It's the troponin. Why? It has 3 binding sites. It is the binding site where the calcium will bind. It has what? The troponin C. It has also the troponin T. It also has the troponin R, which is the inhibitory. So this structure I drawn here. That's what I'm saying about the troponin, the key protein molecule for contraction. The key protein molecule for contraction is called what? It's called the troponin. Okay, another thing, there's this correlation. I know people are having a slow connection, but here there will be a recorded video there. Okay. So there's another protein. It's not on the sarcomere, but you have to know this. It's called the dystrophin. So do you know what this is? So this dystrophin is one which links the external lamina of the skeletal muscle to actin filament. It links external lamina of skeletal muscle to actin filament. Now, I want you to understand what will be the absence of this dystrophin protein. The absence of this dystrophin protein will result in what is the correlation, what we call the Duchenne's muscular dystrophy. And they like to ask about this correlation in physiology, both in physiology and anatomy. You have to know this correlation. And there is an assignment I want you to do on this same thing related to muscle contraction. Okay, and that correlation doesn't miss in test 1 physiology. So you have to go and research. I'll give you. So this is called the absence of this dystrophy. Absence. I'm saying the absence is to be called what? The Duchenne's muscular dystrophy. So if you want, go and dig deep and research on this, how it comes about and all that. Okay. Now the sliding filament, the most important thing during muscle contraction. So during muscle contraction, what will be happening to the sarcomere? What will be happening to this one? what will happen during muscle contraction? So there will be overlapping of the thick and thin filament. Overlapping will increase, like they will be sliding each other like this, you see. So this head, then when the active binding site has been exposed, this will come and bind here. So they will be pulling each other, sliding on each other. So when they slide on each other, the length of the sarcomere shortens, like the discs, like they will be coming at the center, or the central M line gate. Like here, there will be the length of the sarcomere will shorten. Now, what about the length of the I band, which is the isotropic, the light band? So this light band also shortens. This region also gets shortened. It gets shortened because it's being put towards the M line, the myomesin. Okay. So what about the dark band, which is the A band? So the dark band, which the A band doesn't change. This thick filament region, the region of the thick filament doesn't change. Okay. But the H band narrows because of the encroaching thin filaments. Okay. So let me just give you these pointers. Okay. So pointers. Number 1, during muscle contraction, there will be overlapping of thick and thin filament. They will increase, the overlapping will increase. Okay. 2, the length of the sarcomere shortens. 3, the length of the I band, or the isotropic light, which is the light band, shortens. Okay. 4, the A or the dark band do not change. So these are the important things that you need to know. Okay, during muscle contraction, what will be happening to the sarcomere? You have to remember this. They usually ask, Dr. Demola usually asks this. Okay. Now let me give the last pointers and we close the video. Due to technical challenge, you having slow connection, we cut the video like that. No. So let's just me give you the pointers on cross-bridge cycle. There are just 5 steps on cross-bridge cycle. Number one is attachment. Okay. Attachment. So attachment, that's where the myosin head you attach to the exposed site of the actin filament. Okay, that's the number 1 point. Number 2 is release of myosin head. The release of myosin head. Release of myosin head. So like, how is this myosin head going to be released? So that the ATP will bind with the myosin head, and it results in release of myosin head from actin or thin filament. Like the ATP, when it binds, it releases the myosin head. So the myosin head gets back from its normal position. You see? Yeah. Okay. So number 3 is bending of myosin head, or which is called the recovering stroke. There will be bending of myosin head, which will be the recovery stroke. So the conversion, this is why there will be conversion of ATP into adenine diphosphate, the ADP, and the inorganic phosphate. This causes the rotation of myosin heads. Hence, the rotated myosin head will assume pre-power stroke position. Okay, and then the stage 4, that is stage of power stroke of myosin head. That will be stage stage of power stroke or what? Mousing head. Okay, I'm just giving you a summarized point here, but I've explained deep into the in the tutorial document. I'll share the tutorial document after the video. Okay, there will be stage of power stroke of mousing head. I'm sure you're able to see my handwriting there. Okay, so the stage of power stroke. What is happening there? So on the rotation, the mousing head reaches near the new binding site of actin filament. So meanwhile, the mousing head will bind weakly with thin filament at the new binding site. Hence, there will be release of what? Inorganic phosphate, ADP, then form mousing head. Mousing head causes what? The straightening of mousing head and movement of thin filament, or the striding. And that is what is called the power stroke. Okay. And the ADP will get detached from the mousing during power stroke. So the point that you need to remember, during power stroke, the ADP will get detached from the mousing during power stroke. Then the stage 5, the last stage of is the stage of reattachment. So the mousing head from the tight bond with the which will bond with the actin till the attachment of the ATP. That is like the cycle start again. So all this happens, produces movement of the thin filament over the thick filament. Okay, so what's just moving there is the thin filament. The thick filament are not moving. Okay, now let me give you the assignment here I was talking about. So here's the thing I want you to go and research. How is myasthenia gravis caused? How does this comes about? And they actually just give you 2 marks for this. It's very simple. This came in test 2, in test 1, 2024. So go and search. You need to submit in my inbox. If you have watched the video, you need to submit in my inbox. I see what your answer is. Okay, thank you very much for those of you who have persevered to the end of the session. Yeah, otherwise have a blessed evening, and don't forget to study and pray, right? So we'll give more pointers on muscle contraction, and it will be explained in the next